In this module, we will talk about the fine structure of DNA. My hopes for you are that you will understand the fine structure in these following aspects. First, we will speak about the fact that the DNA strands are themselves polarized. We'll talk about how polarized strands interact to form the double helical structure. We'll speak about the nucleotide bases that mediate the interactions between the two strands. We'll talk about the pairing of the DNA base pairs, how the pairs are formed, and how the specificity is behind the base pairing. The first thing that I want to have be apparent to you is that, like all polymers in biology, DNA is a polarized polymer. One end of the polymer is chemically distinct from the other. We distinguish these two ends based on what is attached to the carbons of the ribose sugar. The 5' end, shown toward the left-hand side, has a phosphate chain attached at the end, and the 3' end has simply a hydroxide attached to the ribose sugars. The individual nucleotide bases, which are on the 1' sugar shown at the top of the picture, all orient in the relative same direction with respect to the polymer. When the DNA is created, new nucleotides are added only onto the 3' end. DNA, as it exists in cells, is formed not of one polymer but of two polymers. The two polymers interact by wrapping around each other in an anti-parallel orientation. This anti-parallel wrapping orientation forms what is known as the double helix. The interaction between the two strands is mediated by the base pairs that line the channel between the two strands. The actual base pairs interact by complementary chemical signatures that exist within each base pair. We will now talk about the 4 nucleotide bases of DNA. These bases fall under two chemical categories: purines, which are composed of 2 fused aromatic rings, and pyrimidines, which have a single aromatic ring. The 2 purines are adenine and guanine, and the 2 pyrimidines are thymine and cytosine. When adenine is incorporated into a nucleotide, the nucleotide is called adenosine. When guanine is incorporated into a nucleotide, the nucleotide is called guanosine. When thymine is incorporated into a nucleotide, the nucleotide is called thymidine, and finally, when cytosine is incorporated into a nucleotide, the nucleotide is called cytidine. In DNA and cells, only 2 different types of base pairs are observed. Adenine will pair up with thymine, as shown to the left, and guanine will pair up with cytosine, as shown to the right. The pairings themselves are done by hydrogen bonds that form between each base pair. Notice that on the left, the AT base pair has 2 hydrogen bonds illustrated with dotted lines, while on the right, the GC base pair has 3 hydrogen bonds. Notice also that the ribose moieties are not found on opposite sides of the base pair. Rather, they're both found on the same side of the base pair. Thus, we can envision a smaller face of the base pair, shown with the red arrow, or the larger face of the base pair, shown with the blue arrow. If you were a protein that wanted to recognize individual base pairs within DNA in order to bind a specific sequence of DNA, would you prefer to bind the base pairs on the smaller face or on the larger face? In order to understand how the base pairs form and how the forming is so specific, it is best to think of the pairing as a lock and key mechanism. The lock and key mechanism works best when 2 different conditions are held. The first condition is that all partial positive and partial negative charges match up as the nucleotide bases approach each other, and that a proper distance is obtained between the 2 sugars on either side of the base pair. So what we will do is we'll identify the partial positive and partial negative charges within each of the base pairs that form the interface with either ellipses for nitrogen-hydrogen bonds or with circles for the nitrogen or oxygen atoms that have lone pairs. When you have a lone pair of electrons, you have a partial negative charge surrounding the system. When you have a hydrogen bound to either nitrogen or oxygen, because of the lone pairs, the hydrogen will pick up a partial positive charge. So that what you want to do is you want to form an interaction between the partial negative of one molecule and the partial positive of another. We will now go through how adenine pairs only with thymine in DNA. If we look at the structure of adenine, we observe on the top tier that hydrogen is a partially positive atom bound to nitrogen, and on the bottom tier there's only a lone pair attached to nitrogen, which has a partial negative charge. The only base pair that will complement this adenine must have a partially negatively charged atom on the top tier and a partially positive hydrogen bound to a nitrogen in the lower tier. And the only case where this is observed is with thymine. And we illustrate this with the oval. We observe a similar complementarity between adenine and guanine. However, in guanine there's a third tier that has no corresponding pair in adenine, and the two riboses are too far apart to satisfy the requirements for the DNA double helix. In the case of thymine, which has an oxygen with lone pairs and a partial negative charge on the top tier, and a hydrogen with a partial positive charge bound to nitrogen on a lower tier, its complementary nucleotide base must have a partially positive hydrogen atom on the top tier and a partially negative charged atom on the lower tier. The only base pair that satisfies this requirement is adenine, and this is shown again with the oval. In the case of cytosine, although the top two tiers do have a charge attraction, on the bottom tier there is an oxygen with a partial negative charge that would not be attracted to the oxygen with a partial negative charge in thymine. In addition, the two riboses between thymine and cytosine would be too close together to form a proper DNA double helix. Guanine has, on its top tier, an oxygen with lone pairs and a partial negative charge, and on the bottom two tiers, hydrogens with partial positive charges bound to distinct nitrogen atoms. Its complementary base pair must have, on its top tier, a hydrogen with a partial positive charge, and on the bottom two tiers, nitrogen atoms or oxygen atoms with partial negative charges and lone pairs. The only case where this occurs is with cytosine, as shown in the lower right-hand corner. Although, in the case of a guanine-adenine base pair, the top two tiers have charge complementarity, the bottom tier in guanine is not satisfied by anything in adenine. In addition, the two ribose moieties are too far apart to allow a proper formation of the double helix in DNA. Finally, cytosine has on its top tier hydrogen with a partial positive charge, and on the lower two tiers, nitrogen and oxygen atoms with partial negative charges. Its complementary base pair must have a partially negative charged atom with lone pairs on the top tier and hydrogens with partial positive charges on its bottom two tiers. The only case where this occurs is with guanine. Thus, cytosine can pair only with guanine in DNA. Although on the top two tiers there is charge complementarity between cytosine and thymine, on the bottom tier the two oxygens with lone pairs would not attract each other. In addition, the two riboses bound to cytosine and thymine would be too close together for this base pairing to be stable in DNA as it exists in cells. So to summarize, each strand of DNA is polarized. The two polarized strands form an antiparallel double helix. Adenine, guanine, thymine, and cytosine are the four nucleotide bases of DNA. Base pairing in DNA, specifically adenine only with thymine and guanine only with cytosine, occurs by hydrogen bonding. The specificity of base pairing is achieved by the proper orientation of hydrogen bonds in each base pair.